I'm very happy to be here today and that you are participating to this webinar. The main idea of this session of webinar is to show you some case studies in which fluorescent ligands have been applied, especially in the field of drug discovery. So as Alessandro was mentioning you before, today we decided to present a work which was made in collaboration with the Institute of Research here in Santiago de Compostela, IDIS, where adenosine fluorescent ligands were used for the identification of new targets for the treatment of colorectal cancer. So just a brief description of our company. Celtarys is a spin-off of the University of Santiago de Compostela, which was created by a group of researchers with a long-time experience in medicinal chemistry, chemical biology, and pharmacology. The technology on which Celtarys is based was developed in the University of Santiago de Compostela, and we apply it to the development of specific and optimal fluorescent probes. These probes are used to facilitate, to be applied in different assays to facilitate target identification, which is the case I will show you today. But also in other phases of drug discovery process, such as hit identification and lead optimization, since they can be applied in fluorescent-based quantitative measurements. So, for example, they can be used to screen molecules and their interaction with the target of interest in the hit compound identification, and also to test the structural modifications that are introduced in the hit compound structure during the lead identification phase in order to increase the effectiveness and safety of such compounds. So our technology is a semi-combinatorial conjugation strategy, which allow us to assemble in a very efficient way polyfunctional compounds. So we have this access to very complex ligand architectures. Thanks to our methodology, a pharmacophore A, as you can see on the right side of the slide, can be easily conjugated to, for example, a fluorophore B or another tag in bivalent but also multivalent architectures. So here you can see an example of the general structure of a fluorescent ligand. So A is the pharmacophore and B the fluorophore, and they are conjugated by a linker that we generally separate in different parts, so a spacer and two hinges. We do that because in our technology, the linker is built in the moment of the reaction. So basically, we assemble different parts, so taking them from different toolboxes. So we use our ready-to-use internal libraries of toolboxes, where we have, for example, 20 different hinges and 20 and spacers. And so you can understand that these hinges and spacer can be assembled in hundreds of ways. And so we have access to hundreds of different linker structures. And so this allows us to tune the properties of the linker, like length and physicochemical properties. But also, thanks to the convergence of the technology, we can assemble libraries of compounds where we, for example, use different fluorophores. So in that case, we are also tuning the photophysical properties of the fluorescent ligand. So basically, we have access to a very wide chemical space. In addition to efficiency and convergency, this technology is versatile. Indeed, it is compatible with most of the chemical structures of pharmacophores, linkers, and spacers and hinges, and it is also environmentally friendly. friendly. It is also important to remark that this company was funded by researchers with expertise in medicinal chemistry. So in all this process, we also apply the know-how in medicinal chemistry, especially when we have to select the pharmacophore and we have to modify the structure of the pharmacophore in order to develop the fluorescent tool. So the colorectal cancer represents the 10% of the annually diagnosed cancers. The majority of these CRC are sporadic, and 20% of patients have metastatic diseases. In addition, CRC is highly heterogeneous and shows high intrinsic variability. So considering this background, it is easy to understand the importance in developing new tools which allow to develop proper CRC molecular classification strategies for patient stratification, and also to deepen into the characteristic of the CRC in order to be able to distinguish between the different subtypes and identify the best therapy to be applied in each case. So GPCRs have been reported to be involved in tumor development, but just a few drugs target GPCRs in the treatment of cancer, and it is due to the lack of optimal target identification strategies. For example, a GPCR that is involved in colorectal cancer is the CXCR4. Indeed, this receptor is involved in tumor growth, invasion, angiogenesis, and metastasis, with a clear prognostic impact for the patients with CRC. Another very interesting target in the treatment of cancer is represented by the A2 adenosine receptor. The activation of this receptor promotes the angiogenesis in a tumor environment, also increases the proliferation of cancer cells, and in addition suppresses the immune response to cancer. The A2B receptor has a very low affinity for adenosine. So basically, it is activated when very high concentration of adenosine are achieved in the medium. So, and it happens basically during inflammation and in the tumor microenvironment. So, in the Health Research Institute of Santiago de Compostela, they decided to perform a gene expression pilot study of colorectal cancer cells, where they studied the gene expression of 92 GPCRs through a real-time quantitative PCR, considering 3 different colon cancer cell lines. So the HCT116, the SW480, and the corresponding lymph node metastatic cell line SW620. So just 36 of the 92 GPCRs showed a detectable signal, and they are reported here in this figure. And after the study of the gene expression in cancer cells, these data were compared with the gene expression in cancer-associated fibroblasts, which is the vast majority of cells that are found in the tumor microenvironment of the colorectal cancer. So as you can see, it seems that 7 GPCRs are CAFs specific, so they are specifically expressed in CAFs, and 10 GPCRs are cancer specific. So for example, you can see here that we find as cancer specific the CXCR4, that is the receptor I was mentioning you before. Other examples are also the cadherin receptor and the adenosine receptor, A2A and A2B, while, for example, the A1 seems to be CAF specific, and the A3 is not reported because it seems to not be expressed neither in CAFs nor in cancer cells. And another example of CAF specific GPCRs is the family of atypical chemokine receptors. So as you can see that there is an unequivocally cancer-specific expression pattern for the A2A and A2B receptors. So based on this data, we decided to develop fluorescent probes to study the adenosine receptors. So you can see here the CELT327, which is an A2B/A3 adenosine receptor fluorescent ligand, and CELT228, which is an A3 fluorescent ligand. The data reported here are obtained by radioligand binding competition assays. So the percentages are percentages of displacement at 1 micromolar concentration. When this percentage is higher than 50%, the Ki is measured. So this is the case of A2B and A3 for the 327 and the A3 for 228. So both fluorescent probes are antagonists, and they are excited and emit in the region of the orange region of the spectrum. The first assay in which CELT327 was applied was the study and the labeling of the A2B receptor in CRC cells. So you can see here this first experiment, which was performed in order to identify the best concentration to be used in the assays. So you can see that how increasing the concentration of the fluorescent probe, fluorescent signal increases, then there is a saturation between 500 and 1500 nanomolar. In the end, it seems that the optimal concentration to be used is 250 nanomolar, because at this concentration there is a well-detectable fluorescent signal, and also because it is always better to not use very high concentration of fluorescent probes in order to avoid out-of-range saturation artifacts. We also studied the specific binding. So in the same concentration, a competitor of so a molecule that binds the same target of CELT327, so the A2B receptor, is added. So they compete for the same binding site. But in this case, MRS is added at a higher concentration. So in this case, the CELT327 is completely displaced, and there is no residual fluorescence. So we can say that the signal is due to the specific interaction with A2B and not other structure, cellular structure present. After that, at the same concentration, so 250 nanomolar, CELT327 was used to label the other two CRC cell lines that I mentioned you before. And you can see that if you compare the fluorescent signal with the data obtained previously by gene expression, they are correlated. So basically with this tool, it was possible to perform a semi-quantitative expression analysis. As I was mentioning you before, A2B is a cancer-specific GPCR, and in order to confirm this data, the fluorescent probe was used in a co-culture of CRC cells, in this case HCT, and CAFs. And you can see that the fluorescent probe, that is a red-emitting fluorescent probe, is labeling only CRC cells and not the fibroblasts, which were labeled with cell tracker. So in order to further demonstrate that and confirm that the A3 receptor is not expressed in these cell lines, CELT228 was used, which is as A3 fluorescent ligand, to label, to study these cells. So as you can see, there is no fluorescent signal. So basically, these cell lines are not expressing A3. And in order to have a positive control, CELT228 was used to label HeLa cells which stably express A3. And so you can see here the fluorescent signal. Another assay in which CELT327 was applied is a real-time dynamic labeling of the A2B receptor in CRC cells. So at 1 minute, there is a detectable signal of a detectable fluorescent signal. At 2 minutes, it becomes evident, and at 10 minutes, the fluorescent signal is saturated. And you can see also that after 20 minutes, there is a slight reduction of the intensity of the signal, and it is due probably to some photobleaching. So basically, CEL327 labels in a stable and efficient way the cells. You can see also that the labeling is uniform, because if you check this orthogonal view of the image, you can see that the fluorescent probe is labeling also both the apicobasal and the lateral side of the membrane. Considering that the field of marker identification is trending towards complex tissue-like cellular systems, because they mimic more faithfully the tumor environment, in vivo tumor environment, we decided to test this CEL327 also in spheroid models. It is important to remark the usefulness of fluorescent ligands in this kind of assays. If we think that, for example, antibodies show a low penetrability in multilayers of cells. So using fluorescent antibody, you could lose a lot of information. So as you can see from the video, the fluorescent probe seems to label uniformly the spheroid, differently from cell tracker, which with it was compared on the left side. And it's more clear if you see these two different graphs. So here on the left side, we see that the fluorescent signal of CEL327 keeps CEL327 keeps a good fluorescent signal going into the spheroid, while for example, cell tracker loses completely this signal, so because it has a very low penetrability. And on this other graph, so at the diameter of 60 micrometers, you can see how the fluorescent signal of CEL327 seems to be uniform on the entire diameter of the spheroid. Another study we performed was the study again of the specific binding, but in this case with a real-time monitoring. So after the addition of the competitor, MRS 1220, the fluorescent signal decreases. And as you can see also in this graph on the right side, and we are sure that this reduction of the fluorescent signal is not due to bleaching of the fluorescent probe, because in this experiment a control was used where only CEL327 was monitored without the addition of MRS. And so you can see that there is only a slight reduction of the signal for some bleaching. So basically this loss of fluorescent signal is due to the competition with MRS. So thanks to all this characterization of CRC cell lines, we can say that A2B is a potential target for the treatment of CRC. So in this case, a preliminary study was performed, treating the CRC cell lines with A2B and A3 antagonist. So in this case, the two pharmacophores used are ISAM 140, which is a specific and selective A2B antagonist, and ISB74, a selective A3 antagonist. So as you can see here in the histogram on the right side, the antagonist of the A2B receptor induces a reduction of the proliferation of CRC cells, while the ISB74, so the A3 antagonist, does not reduce the proliferation, because basically it confirms what we observed previously, considering that the A3 receptor is not expressed in these cells. So just to a brief summary of what we were showing you today, the gene expression study in CRC cell lines allowed you to identify the A2A and A2B adenosine receptors as CRC cell specific expressed. Cell 327 and cell 228 were developed to study CRC, and they seem to label cell membrane in fast, stable, and specific manner. The cell 327 was used in different experiment setup to perform a semi-quantitative receptor mapping in living cells, in real-time A2B receptor labeling, and also for the labeling of cellular systems of increased complexity. And the treatment of CRC with the A2B antagonist ISAM 140 revealed a reduction in the CRC cells proliferation, so confirming the previously observed data. So to conclude, we can say that fluorescent GPCR ligands are useful preclinical tools for the comprehensive characterization of GPCRs in live imaging modalities. So putting the basis for the design of new expression studies, drug screening platform, ligand binding interaction analysis, and also that Celtarys technologies allows to identify optimal tools for different targets, because here we have examples of GPCRs, but we are validating it also for other kind of targets, and to obtain tools suitable for different application in a very efficient manner, trying to make fluorescence the new gold standard technology in drug research. So I would like to thank my colleagues in Celtarys, our collaborator who did all these biological assays in the EDIS Research Center, so Dr. Jorge Barbasan and Miguel Aval Posada, Professor Sotelo Perez and the Combiomed Research Group, the SICUS Research Center, the InnoPharma Group, and also Xunta de Galicia, Bioincubatec, and Programma Ignicia for the financial support. And thank you for your attention, and I'm here to answer your question if you have any question.
Thank you, Maria, for the presentation. Super clear. And again, if you have any question at the moment, don't hesitate to ask. But I know that maybe it was quite intense seminar, a lot of information. So if you have any other questions, technical or other kind of question, don't hesitate to contact me or Maria. At the end of the meeting, we will send you the video. So the video was recorded and you can have access when you want. Okay, so if anyone else have any question, thank you very much. And I hope you have a good resting of the day, and see you next time. Grazie, Maria.
Bye-bye. Thank you,
Bye.
everybody.
Bye-bye. Thank you. Bye.